All right, welcome back, everyone. Can you hear me okay?
Yep, we can hear
Thank
you.
you. Thank you. I just got out of a Zoom meeting, so I had to switch over here real fast, and I was afraid that it was going that maybe something was gonna go wrong. Technology.
Yeah, so far so good.
Okay, thank you. Let me know if it goes away.
Sure.
All right. So as you probably already know, this is going to be our last lab meeting, and then what you have next after this is going to be to turn in your mix unknown, and also you have your last lab test, which is the unit 4 lab test, which includes the fungi, includes the protists that we went over last week, and then what we're covering today, the helminths. Now, you probably remember that this word helminth is something that we use to refer to anything that resembles a worm. All right, so just like before, we will go over them and we'll try to find maybe some specific characteristics that we can use to help differentiate among some of them. Personally, I feel like it's a little easier to differentiate among the helminths that it may be to differentiate among some of the protists. So hopefully you feel the same way. If not, just ask. Like for example, I remember it may not have been last week, but Joe, for example, in one instance specifically asked, what can we use to differentiate this from that? And I'm happy to try to come up with something for you. All right. So we are going to start with some of the flukes. So remember, there's two types of flatworms. We have the flatworms that are not segmented and the ones that are segmented. So the flukes are going to be those in the category of not segmented. And the first one that we'll look at is Schistosoma mansoni. When a patient presents with Schistosoma mansoni and we look at the stool of the patient, more than likely what we're hoping to find there is going to be, and I'm switching color in my pen right now, this egg sacs. What is very distinctive about these egg sacs is over here at the end. This is spine, and notice that that spine is not in the center. It's actually kind of like on the side of the egg sac. This is something that we will use in combination with the patient's symptoms to get to the final diagnosis. Now, you probably remember seeing in some of our Unit 5 lectures something that resembles this, and this is going to be the free-living, at least temporarily free-living, larval stage for Schistosoma mansoni. This is the one that swims towards you. That's why you have this structure here. More than likely, you will not be able to find this in a patient sample. This is something that you would see swimming around in the fresh water. And then attached to organs in the patient, maybe to the liver, we will find sometimes the adult stage. Sometimes we find them also in the intestinal area. And notice though, in this picture, the adults, there are two of them here. We have the male and the larger one that looks kind of pink here because of the stain that was used is going to be the female. The way it works out with the helminths is that the females tend to be larger than the males. And in this specific case that you have both of them in this slide, it's because they're actually fertilizing eggs at this point. Now, the next fluke that we're looking at, in this one, you do not have female and male. You only have one type because they happen to be the types of flukes that can carry both sets of reproductive organs. What I want you to notice on the left-hand side, the egg again. Here you have another egg sac, and you're probably thinking, well, that is very similar to the egg sac that we just looked at. Not really. The Schistosoma manzoni egg is a lot more elongated, a lot more translucent. And even though it does have a spine, the spine is actually not sharp, it's actually blunt. And more importantly, besides having that blunt spine, notice that there is this structure at this end. This is actually what we refer to as a knob in the egg sac. Now, the adults that may be found stuck to liver tissue are going to look like this. And the reason why you have the different colors here, it has to do because this specimen was fixed and it was stained on the slide so that you could see more contrast. So you can see there are both sets of reproductive organs. Female reproductive organs are going to be right by the intestinal area. There's ovaries here, and then we have the male reproductive organs that are going to be very close to the excretory bladder. Are there any questions so far? All right. Now we do have one more fluke to go over, but notice how this Clonorchis sinensis shape is kind of wide, and I always say that it's an oval shape. The Schistosoma manzoni, they were elongated. When we look at Fasciola hepatica, here you have the adult. Notice that the shape of the adult is very different from both the shape of Clonorchis sinensis and the shape that we discussed or that we looked at for schistosoma. Now, there's also only one version of the adult here because they have both sets of reproductive organs. These adults may be found in intestinal area, may be found in liver tissue. The eggs, as you can see here, do not have a spine like we saw for Clonorchis and we saw for our first one, schistosoma. And you do see a knob at one end. Are there any questions? All right, thank you. So next we're going to look at other types of flatworms. These are the cestodes which are segmented. Now, before we go through the different ones that we're going to examine, notice that depending on which type of tapeworm we're looking at, we may have different sizes and shapes of segments. It is a lot easier to identify tapeworms if we find the segment that is basically the equivalent of a head. It is not a head, that we refer to as the scolex. Scolex usually have within them structures that allow them to stick to tissue that we refer to as suckers. Makes sense? They're kind of like plungers that they can use to stick to areas. Sometimes they may have what we call hooks. Some have hooks, some do not. So here you have an example of one where you see that the segments here is segment number 1, number 2, number 3. This one doesn't have, first of all, Echinococcus granulosus doesn't have all that many segments. That's the first thing that you need to notice. The second thing that you can see here is that the segments seem to be of different size and shape. If you look at the second segment here, it's a lot more rectangular than the third segment, which tends to be a little bit more of an oval shape. Here you have the one that we refer to as the scolex. These are going to be the area where we have those suckers. So Echinococcus granulosus is going to be one of those that we sometimes see in patients, stool samples. And it may be just because, you know, they may have ingested egg sacs, or maybe just put their dirty hands in their mouth. That's usually what we see. But more than likely, if you're going to see this in real life, face to face, it's probably going to come from a pet, like maybe a dog that has it in their stool, just to give you some perspective on this. I'm not going to ask you anything about the disease per se, but that may give you an idea of something to use to remind yourself of that shape. All right, the next segmented worm or tapeworm or cestode that we're going to look at is Taenia pisciformis. There are several species of Taenia. Taenia pisciformis is one of the smallest ones as far as the size of the scolex and the size of the segments. I'm going to try to make a circle. Hopefully I'm successful here. Sometimes I lose control of this pen, of the area that we're going to refer to as the scolex for Taenia pisciformis. Notice that there are some suckers here, and I know it's very difficult to see them in this picture, but there are these ridges. All of those are going to be very small segments for Taenia pisciformis. Now, if we look at the eggs of Taenia, especially the Taenia that we tend to see in humans, whether it is Taenia saginata, which is the beef tapeworm, or Taenia solium, the eggs are pretty much identical to one another. And you may read online where they're trying to tell you that you can actually differentiate eggs of Taenia on microscopy. Very unlikely, I can tell you that for sure. Unless you see the scolex of the organism, all you can say is that there were Taenia egg sacs in the stool of the patient. It takes very specialized type of microscopy to be able to say is one over the other just on the egg. So all of the Taenia eggs are going to have around them what looks almost like a jelly-like structure, unless they have been desiccated completely. And then you can see that they have a very dark inner shell that is actually pretty tough, and that makes it so that those eggs can stay viable outside of the host body for an extended period of time. But again, you are not able to differentiate between the different Taenia that we usually see in humans just by finding eggs. You know there's a Taenia, you just don't know which type. Now, the one that we do want, and really finding Taenia eggs in stool can be pretty serious. You want to know which type it is, because the prognosis for Taenia solium tends to be much worse than it is for Taenia saginata. So if somebody has Taenia solium, they can develop a disease that we refer to as cysticercosis, and that is where they actually can leave the intestinal area. They can get to muscle tissue. Here you have the cysticerca or the cercaria in the tissue of the patient. Usually you find it in brain tissue. Sometimes you find it in some muscle tissue. And because of that, it is very important that we figure out if the patient does have Taenia solium versus Taenia saginata. Taenia solium, here you have the scolex, has 4 suckers, but more importantly, it has 2 rows of hooks. That is going to be very different from what we see with Taenia saginata scolex, where there will be no hooks. Now, the segments are all going to be the same size each other. And here you have just a depiction of one of the Taenia solium segments that we also refer to as the proglottids. Which carry usually tons and tons of fertilized eggs within them. And that's why breaking off a segment basically just releases eggs in the fecal matter of whoever it is that is carrying that Taenia in their body. Are there any questions so far? And again, let's think of size here. Taenia solium and Taenia saginata are a lot larger than the Taenia pisciformis that I showed you earlier. So we're going to move on to the roundworms next. And the first thing before we go into our examples of roundworms is that there are tens of thousands of different types of roundworms out there in soil, moist soil usually, in freshwater environments. There are some that are in marine environments. Only a few of them are going to be able to infect people or animals. And one thing that you need to think about is there are going to be many different sizes of this. So we're going to start with the one that we think is the smallest one that we tend to see diagnosed in the United States the most, Enterobius vermicularis, which is commonly referred to as the pinworm. So if you take stool sample from a patient and transfer some of the worm material into a slide, the adult is going to look like what you see here on the left-hand side. Now, the main difference between the males and the females is going to be size, really, and the fact that we will see that the females are going to be, like I said earlier, larger, and the males have a hook that they basically use to latch on to the females so that they can fertilize those eggs. And talking about eggs, once those eggs are released, they look something like this under the microscope. They're very small in size. This is a very magnified picture. And luckily for us, if somebody does have an infection with Enterobius vermicularis, uh-oh. Oh, so Matthew lost me. Oh, shoot. And he is gray, so he should be okay. He's probably going to try to get out and come back. So luckily for us, what I was getting to is that you will find a lot of these shapes in the stool. Because if it was up to you finding one of these, it's going to be very difficult. So these eggs, in combination with the adults, are going to be a good way of diagnosing a patient. Now, to be fair, more than likely, you don't have to even go to the doctor because you know what pinworms are and you can buy over-the-counter medication to deal with the infection. All right, something that you may see presenting in patients, especially as patients that show up with some muscle cramping and some malaise. If you do a biopsy, if there's a biopsy of tissue, specifically muscle tissue, you may encounter in that patient something that looks like this, or here, or here. Notice how many there are here in this sample. These are the larvae of Trichinella spiralis. I always say this because it may help you remember. So notice how they look like pigtails in tissue. These are the ones that you get from eating undercooked pork meat. They look like pigtails in your muscle. they came from pork meat usually. Are there any questions so far? All right. Next, we have something that is actually really easy to differentiate from the rest of the roundworms. And it has to do with the fact that they have this very long, very skinny tail that looks like a whip. They are commonly referred to as whipworms. The genus and species for this is Trichuria, and the eggs are very distinctive because the eggs have two knobs. The egg sacs have two knobs, one at each end. Now, usually when the patients have this, and notice that you have these two eggs side by side here, there's going to be a whole lot of eggs in that sample. That is going to make it very easy to diagnose the patient from looking at that slide. And last but not least, we have one that's a lot larger in size when they're adults, Ascaris lumbricoides. Now, the female is going to be always the larger one. Notice how the male, once more, has that hook at one end that is used to help latch onto the female for fertilization. Even though they can get to be almost a foot in length when they're adults, that's the maximum size. The eggs are actually quite small, and they look very similar to the eggs of Enterobius vermicularis as far as size and look to it. They're kind of like see-through on the slide. So again, the combination of the adult organism and this egg shape in stool can help you identify the microbe. I'm sorry, the microscopic part will be the egg, so you can diagnose the patient. Now, for this week, when you go to do the lab assessment, you'll notice that there are a lot more questions because we could maybe think about what type of egg is this? Is this an adult, or is this an egg, or is this a larval stage? Is this a roundworm or a flatworm? What type of roundworm is this? So basically, like what we have been doing since we started with the fungi, these lab assessments should be basically practice questions for your upcoming lab test number 4. Now, as far as reviewing for this lab test, what you need to do is look at the PowerPoint that I posted for fungi, the PowerPoint that I posted for protists, and today's PowerPoint that you have access to in Blackboard, and you're going to try to quiz yourselves. So it may be very helpful for you to actually go to the lab assessments. If you miss anything, make sure that you understand what that was that you missed, so that if you were to see a similar picture in the Unit 4 lab test, you don't make the same mistake. Are there any questions so far? Of course, then it comes that you actually do have to study them. FYI, I'm not going to ask you to try to type, I should say, to type these names in the test. More than likely, well, it is the way it is. You're going to have to choose something rather than feeling something, because I know and I understand that autocorrect sometimes messes you up. And even if you had the best of intentions of typing one of these names, it is very easy for something like Trichuris to get changed into Trichinella or something else altogether. But again, Abigail, I just want to make sure that yeah, you have to be able to recognize them for sure. And I don't necessarily want to change the way you study for this test just because you're not going to be typing them yourselves. So make sure that you're able to differentiate those names that are very similar, like Trichinella and Trichuris for sure, so that you don't trip yourselves up. Yeah, lumbricoides. I know. Well, I mean, there are some deuces here, so I get it. Like with Trichuris, Trichuria, we barely pronounce that second I. So you may not want to write that down, or you may not remember to type it down. All right, I'm going to switch documents so that we can go over the rubric that I posted for the mixed unknown. All right, so a document that you have available to you, and you may need to scroll down, of course, in this week's lab folder, is going to be this rubric. Now, what you have here is the maximum number of points that you can get without any mistakes, or at least following the instructions for each part that you're required to have. Notice that there are some point differences here between the maximum number of points that you can get for the unknown form, for the paragraph, and dichotomous key compared to the actual correct ID. So the assumption that I'm going to make, and the way we are going to go over this, is by assuming that we are shooting for the maximum number of points. So in order to get 10 points for the unknown form fill out for the gram-positive portion, you have to include all the tests that you read. That includes the observations, result interpretation, and you're not adding any extra tests. That can get you up to 10 points. For the unknown form portion, that's the gram-negative portion, you can get up to 10 points if you include all the tests that you read for your gram-negative with the observations and result interpretations and no added tests. Are there any questions about those 2 points? How would you have added tests? Abigail, I am afraid to answer it because I don't want to give anyone any bad ideas. But I know why you're curious. How would you have added tests? Well, what some students have done in the past is that they go online, and if they think they have a certain bacteria, let's say that they think they had Shigella sonnei, an example of a gram-negative, they will go online and find every possible test result they can for Shigella sonnei, whether or not they read those results in the lab themselves. And that is not what we're doing. Yeah, I know. You're probably thinking, why would I do that? Well, some people have done it in the past. Even though the instructions said just to include what you test, what you read every week, but it happens. It has happened. Remember, the idea is not to use every possible lab test that you learned this semester to identify something. The idea is to have a very directed, very short way of getting to a final answer. All right, the next 2 lines are related to the paragraphs. Now, the description of the paragraph, you have it in the instructions in the virtual mix unknown document. But if you include in your reasoning 4 tests minimum or more, you will get the 10 points for the gram negative and the 10 points for the gram positive. Now, remember last week we talked about this. We talked about, because some of you were concerned with how big do these paragraphs have to be. And I said, I don't like to tell you which numbers of words you have to have, because you're all reading different collections of tests. And this is supposed to be how you're reasoning and you're thinking about it. So the way you think about it may require that you use 50 words, and for somebody else, it may take 100 words. So I don't give you a maximum or minimum number of words. I just want you to include 4 tests minimum to get the 10 points. Now, the dichotomous key for the gram negative and the dichotomous key for the gram positive can get you up to 10 points if you set it up correctly. If you have a mistake, then you start losing a few points, and you can see how we keep losing points down the line. Are there any questions so far? And remember what we talked about it, even if, last week, even if you start, and I'm gonna draw something here, let me switch to black. Even if you decide that you wanted to have one giant dichotomous key, where on one side you have positives and in the other side we have negatives, I'm gonna grade them as a gram positive one and a gram negative one separate from one another. And if you decided that you wanted to do them separately, so I want to give you some freedom. Remember, I recommended that you start with the catalyzed test where you have positives and negatives, and then you read the next test that more than likely, if it's a positive, it's going to be an MSA. I know I said this last time, but just to be sure. And if it's a negative catalase organism, more than likely, you're going to continue with the BIOL esculin and so on. For the gram negative, I recommended that you start with lactose, where some were negative because they don't ferment, and some were positive, and so on. I will read them, I will grade them separate from one another, and you can accumulate up to 10 points each. Remember, this assignment is worth 70 points total, this project. Now, the last part, which you're probably thinking, I thought that was what counted the most. It does count, but not necessarily for all 70 points. It's going to be whether or not you got the correct ID. So if you get the correct ID for the gram positive, you get a total of 5 points. That includes correct genus and species. If you just get the genus, or if you forget to write down the species, which, believe it or not, has happened, students like to abbreviate a lot. You should not be abbreviating in a project that you're submitting, but students do things like this all the time. You get 4 points. And if you get incorrect ID, that part, you get 0 points out of 5. The same thing for the ID of the gram negative. Are there any questions? Now, depending on the software that you have at home, you may be able to attach your dichotomous key to the virtual mix unknown form at the end as an extra page. The same thing for the paragraphs. Not everyone has the same type of setup at home. So it may be that some of you fill out the form, you create different documents for your dichotomous key and for your paragraphs. You can upload all those into Blackboard and hit submit once, and you can have multiple documents in one submission. So that is completely possible to do in Blackboard. So I'm not going to require that you do it a certain way or only one way. Either way is fine as long as all the information that I need is available and you uploaded it by the deadline. All right. Are there any questions before you go do that lab assessment? Okay. And just like any of our lab tests, remember that you have to use responders. There are 22 questions, not necessarily 22 pictures. And I'm only saying this out loud because a different section asked me this question before, so I figured I might as well just say it in case you had the question and you were not asking it. All right, just as a reminder, please, you should be working on your Unit 5 lecture test that is a take-home that you will upload in Blackboard. You're going to open the lecture test folder and there will be an upload link in there for it. Don't wait until right before it is due to work on that Unit 5 take-home test, or you will have really bad results. It requires that you do a lot of critical thinking. So usually an hour is not going to be enough for you to work on that test. Are there any questions before we adjourn for the day? Well, have a fabulous weekend. It is Friday, isn't it? I don't have weekends, so it's okay. And if I don't hear from you next week, well, have a good summer. I can't believe we're at the end of week 15 already. Thank you so much for joining me.
Thank you. It's been lovely.
Lovely. I like that. I like that. I am glad we're recording this so that I can actually listen to that again and again and again.
You're welcome.
All right. And if any other questions come up, just email me.
Okay, will do.
All right. Bye, everyone.
Bye. Take care.
You too. You're welcome.